So good afternoon, everyone, for the second session on morphogenesis. And now we can have a talk from Alan Rodriguez from the Rockefeller University, and he's going to tell us about Back to the Future avian systems as portal into the 21st century.
Raise the stakes. So yeah, I guess before I thank you so much to the conference organizers and for the invitation. I guess before I get into explaining how we've been using avian systems in the lab, as a quote unquote portal into 21st century biology, I thought it might be helpful to read a slightly provocative quote written about 20 years ago by Carl Woese. And this is he wrote this piece called A New Biology for a New Century, sort of opining on what biology should be in the 21st century, or sort of the fork that it's facing. So I'll just read the whole quote because I think it's very vivid. So he says, in the last several decades, we have seen the molecular reductionist reformulation of biology grind to a halt. Its vision of the future is spent, leaving us only with a gigantic, whirring biotechnology machine. Biology today is little more than an engineering discipline. Thus, biology is at the point where it must choose between two paths. Either continue on its current track, in which case it will become mire in the present in application, or break free of reductionist hegemony, reintegrate itself, and press forward once more as a fundamental. The latter course means an emphasis on holistic, nonlinear, emergent biology. So, I think what he's sort of getting at is that there's sort of a choice. In a sense, molecular biology, molecular reduction has been really successful. We've learned a lot from it. At the same time, the question is there really something more to be done that's truly fundamental in biology? And we'll get to why I think the avian system is a great place to think about those questions. And I think, in addition with the privileging of the molecular level of organization as the seat of explanation in biology, I think there's also been a reliance on concepts that are in a sense based on machine thinking that have been used to organize the richness that we've seen at the molecular level. There's this two-hand, and I think they've been very successful, and we've learned a lot. At the same time, I guess what people like Woese are suggesting is there more to be done? And I think what's interesting is that over 100 years ago, there were people thinking about these questions too, that I think now the molecular evolution has also led us to today. And these people were called the organicists. There's a wonderful book written about them on the right. And this was actually happening around the same time that this, I'm sure you've seen this picture before of these very famous physicists coming together 100 years ago. And they were really coming together to basically make sense of the fact that the Newtonian mechanical paradigm in physics was falling apart. And they were trying to figure out what that made, what sense that made for physics, and what were people to do about it. But at that same time, there were very astute conceptual thinkers in biology who were aware of what was happening in physics and thinking, well, if the physicists are letting go of their grip of the Cartesian mechanical way of thinking about things, shouldn't we also be doing that for biology? And so they really developed a very rich set of conceptual ideas that they people often refer to as the third way, which is not reducing life to being and a non-living matter, but also not slipping into things like vitalism, where you say, well, you can't explain things because it's more, life is not machine, but we can't really explain it because it's unknowable. And so in the lab, I think Amy and I have, Amy Shire and I co-direct the laboratory, have been trying to take this, revive a organic way of thinking conceptually, but use the avian system as a concrete instance of really bringing these ideas and understanding them in the flesh, so to speak. And so we were very interested in organ structure or morphogenesis and how organs acquire their exquisite structure. And I think the avian system is really special because the morphology that develops in avian systems is really, really robust, much more so than foods in the mouse. And so that's really, it's really nice because the signal to noise ratio morphologically is very, very high. And so, when I was taught to think about morphogenesis in graduate school, I would say that the general way of thinking about it was sort of to see morphology and morphogenesis as downstream of molecular patterning, right? So here's an example of something you might find in a textbook where you have basically a homogeneous field of cells. There's some sort of symmetry breaking event happening at the molecular level. You know, in this case, it could be to something like a molecular Turing reaction diffusion network. Then you have difference across the field, and because of those differences, individual cells do different things. And so you basically have this idea of a pre-pattern sort of setting up, for instance, where the follicles are going to develop. And the specific system, I'm sorry, this is not my computer. This sometimes happens when, this is supposed to just be like a basic chicken embryo. I apologize for that. So the sort of concrete sort of system that we're sort of looking at this, this process of morphogenesis is in the avian skin. And in about 2 days, you really have this remarkable transition from a molecularly and sort of morphologically homologous back to one where you really have this really wonderful regular pattern of follicle aggregates, each of which have their own molecular expression pattern. And I think sort of based on the way of thinking that I just outlined in the last slide, the idea is to basically, look for transcription factors, signaling molecules, understand these cascades, and then over time, you'll sort of understand, how the symmetry breaking works. A key experiment that Amy did that sort of actually initiated our collaboration together over almost 10 years ago now, was the following one, where she really carefully looked at both changes in morphology and cell morphology, both in the dermis and in the epithelium, and also looked at the nuclear localization of beta-catenin, this transcription factor that was thought to be, and sort of still is sort of suggested to be the first sort of first marker of sort of molecular symmetry breaking. So once you have beta-catenin in the nucleus of the epithelium, then you start to have all of these signals being released from the epithelium into the dermis. And what she noticed was that the molecular symmetry breaking did not precede the morphological changes, right? So it's not the situation where you have a nice clean morphology, then nuclear beta-catenin, and then these changes. You're always seeing morphological changes happening coincidentally with the molecular symmetry breaking. So that sort of sets into motion the sort of chicken and the egg question. If they're both happening simultaneously, how do you explain that in this sort of molecular pre-pattern paradigm? And the approach we took was to sort of resolve this issue by sort of taking sort of a conceptual reshift, reframing sort of the conceptual way we were thinking about it. And rather than focusing on trying to explain how a molecular pre-pattern could break the symmetry, rather think about how collective cellular self-organization could be responsible for that. At the time, this is probably around 2015, we were very inspired by some very creative theoretical work by George Oster and Carl Baer that they did in the 80s, where they were looking at collective cell mechanics. And this was around the time when sort of molecular biology was sort of taking over development, but they were sort of thinking about things in this sort of collective cell mechanics perspective. And so this, this is not work that we were exposed to, Amy and I, when we were in graduate school in labs like Cliff Taben's lab. But we sort of wanted to sort of push these theoretical ideas, these conceptual ideas forward, and really test them rigorously in an experimental setting. So long story short, I think the sort of key takeaway of what we were to find sort of suggested the following type of model, where collective cell mechanics are able to spontaneously initiate follicle aggregates through the sort of collective behavior of the dermal cells. And as these aggregates start to form, those aggregates actually compress the overlying epithelial tissue, which then through mechanotransduction activates beta-catenin and then sort of kickstarts a suite of signaling molecules that then are secreted back onto the dermis. So this is in a sense, it's a way of thinking about symmetry breaking at a different level of organization than just the molecular one, and one where you're, in a sense, killing two birds with one stone, so to speak, right? You're sort of making the morphology as you're creating the pattern. So patterning and morphogenesis aren't decoupled; they're really sort of simultaneous events. So there were certain key takeaways that we took away from this, and it sort of formed the basis of how we want to develop our lab at Rockefeller. And I would say it's under this umbrella of what we call supracellular morphological self-organization. So the idea is that the supracellular level, in a sense, has generative morphological capacity, right? It's not just that collectives are sort of being instructed what to do on a cell-by-cell basis by molecules, but there are sort of rules of the game at this level of organization that are creative also. And so since they can't be predicted just from sort of this individual cell properties, they kind of have to be studied in their own right. The second very important issue is not only is there this capacity at this other level, but there can also be downward causation, right? So that level can actually change the properties of the microscopic state, right? So it's not just bottom up, but also in a sense top down. And once you start thinking about that, there's sort of many feedbacks that sort of need to be considered. And the final thing is sort of appreciating the sort of really powerful collective capacity of the mesenchyme. I think they tend to be thought of as sort of being these sort of simple, disorganized support cells, but at a collective level, this study suggested to us that they sort of have incredible organizing capacity. So we wanted to delve a bit more into what was really going on with these mesenchymal cells from the dermis. And so we developed an ex vivo system that allowed us to observe this collective cellular self-organization basically ex vivo. And again, another example, I think, of the avian system being wonderful. This is a great system to get really precisely staged tissue, a lot of it, and then also basically take advantage of basically a recombination type setup, right? These cells avian cells seem to be really good when you're culturing them ex vivo. And so this is what you see when you put 20,000 cells down in a ring. We're sort of simulating the midline of the back of the avian embryo, which is how the follicles emerge in vivo. And there's sort of a similar time and length scale. And so basically this sort of allowed us to look at how these cells collectively organize. So these are cells that are taken out of the dermis, they've been scrambled, put here without the epithelium, so they're not getting exogenous signals, and they can still form these nice regular patterns. And so what's going on here, right? So I think the first thing was to sort of appreciate that there's, in a sense, a reciprocal relationship between the cells and the matrix, right? It's not that it's not really about sort of one thing or one factor doing something. It's that there's sort of a reciprocal relationship between cells and matrix, and this dynamic relationship evolves over time. And what basically ends up happening is that the cells of the matrix over time basically start to form sort of an aligned cell ECM composite that basically increases the contractile behavior of individual cells, but then also sort of increases the sort of alignment of the matrix. And this is something that we also saw happen in vivo. And I think this is where we sort of, I think this step was sort of where I think we took a bit of, made a bit of, you could say, a whole holistic leap conceptually, which is that we sort of thought that while things are happening at a granular level between cells and matrix, you could imagine that at the sort of collective level of organization, at the supracellular scale, the composite of the cells and matrix also actually, in a sense, possess certain emergent properties. And so specifically, that in a sense, the collective as a whole, at sort of the collective level, exhibits behavior that's akin to a contractile fluid. And why this is important is that you can basically, in a sense, force grain the behavior of individual cells. It's not that the individual cells aren't doing something; they are doing something in sort of forming a particular type of organization. But the point is that once you get a particular organization, that creates a property at another level, which then can, in a sense, have a life of its own. And so we did, working with a theorist, Anna Erzberger, we modeled the sort of continuum, the cell ECM continuum, as an active attractive fluid, and we're able to show, both through theory and experiment, that you could that in a sense, once you generate this active contractile fluid, that could actually in itself, on that level, spontaneously form these aggregates. So again, there is sort of a multi-level aspect to this, and I think it is, in a sense, you could say holistic. You have these reciprocal relationships at one level between the cells and the matrix that enable, because of the particular organization they adopt, emergent properties at another level. And that emergent level has material and mechanical properties that then can lead to spontaneous patterning, right? So it's not like the individual cells are necessarily being told who to become or where to go, or that they're sort of moving one by one in sort of a chemotaxis type way of thinking, but they're really sort of behaving collectively at that level. And I think another point here is that I think it's, you know, a point that became clearer to us, which is that physical concepts can be used to sharpen sort of an organic perspective or a mechanical one, depending on your particular viewpoint. So far I've sort of suggested this idea that sort of mesenchymal collectives can have self-organizing capacity, given some window into how this collective really functions through cell ECM relationships and creating emergent properties. But then the question, you know, that we used to get a lot, and I think it's a good one, is, well, what are the signals doing, right? If you may not need signals to, you know, tell individual cells where to go or break symmetry, what are they doing? And I think our view of this, which is, you know, informed by the idea that you can basically create new phase properties at another level, is that what morphogens are really doing is to sort of, in a sense, enable a more expansive palette of biophysical supercellular properties. And so you can basically start to then get sort of, in a sense, a polyphasic system that can, in a sense, do morphological work. So the two signals that we focused on, and this is sort of informed by a lot of the work that's been done in the skin, is BMP or BMP and FGF. And what we found is that FGF really serves to make dermal mesenchymal collectives more solid at the supercellular level and less mechanically active. Whereas BMP makes the collective more fluid and less mechanically active. So first, I'll just sort of give you a picture of some of the experiments we did to sort of come to these claims, and then explain what's the purpose of that in the context of skin morphogenesis. So you know, this area of inferring mechanical and material properties at the supercellular level is not, you know, as established as sort of characterizing things at the cellular level. So a lot of the times we are sort of trying to be creative with making these measurements. One assay that we came up with actually by accident when we were doing SEM is realizing that SEM preparations can actually crack a dense dermal collective. We wanted to look at these under SEM, and then we noticed that there are these cracking patterns. And in material science, people use cracking patterns to infer things about the material properties of a material. So in this case, and so if you get big, large cracks, like when you see in a desert, that's sort of indicative of something that's more solid-like. And so we saw when we treated these collectives with FGF, we get these sort of large cracking patterns. And with BMP, they actually become finer than what you see in control. So this is suggestive of the fact that FGF is generating a more brittle, solid-like tissue. We also did work where we sort of work, another sort of type of experiment you can take spheroids. You know, if you take two materials and you sort of put them next to each other, you can imagine if they're a fluid, they would sort of coalesce into one big drop, whereas if they were sort of hardened, they would sort of not do that as much. And so here you can see when we treat the dermal collectives with BMP, it in a sense fluidizes things, and they form an aggregate sphere much more quickly. So BMP appears to be generating more fluid-like properties. So now this is something that I think was a very exciting experiment for me, which is characterizing these properties in vivo or in a section of a tissue. And you know, one key fact that we learned through the course of this study is that there's an inner core. Once the aggregate starts to form, there's an inner core that's FGF signaling positive, ERK positive. And then there's this margin that goes around it that's actually phospho-SMAD positive, indicating BMP signaling. So in a sense, this is sort of FGF, you know, FGF is working here and BMP is working here. And we used atomic force microscopy to probe the stiffness of these two different regions. And when we used a small probe that's sort of the size of an individual cell, we didn't see much of a difference. But when we used a bigger probe that sort of scans, you could say, sort of a supercellular length scale, we were able to see a more significant difference, right? So this is really, in a very tactile way, it's really sort of indicative of, in a sense, an emergent phenomenon, right? Emergent, sometimes people think of as sort of this woo-woo concept, but you can physically do it, right? You can, on a more granular level, not see a difference, but then on a larger level, because of the organization of those components, see something that emerges as different. And so now when you have these two phases, you can basically, in a sense, make compartments without a basement membrane in between them. Working with an applied mathematician, Pearson Miller, we argued that, in a sense, the contractile phase around the margin basically works to propel the more solid core of that's actually posited out to sort of allow for budding. And again, I think the key point here is that this is really happening on a level of organization that's not the individual cell level, right? So it's a bit different than sort of migration of individual cells. And so it is a different, I would say, a different perspective that may be worth considering in other situations. So I think the key takeaways here was just, in a sense, that by accounting for the supercellular level organization, I think we realize that one may need to distinguish between, in a sense, the proximal effect of a morphogen or a signal, which is obviously a change gene expression, and the sort of functional effect, which really emerges at a different level of organization that you have to characterize to even know of its existence. So I think I'll end with this final picture. This is sort of my very, very simplistic, the way in which my thinking of development has really evolved. I think when I started graduate school, I thought about development like this, when I was trying to reprogram limb progenitors. And now I think about it more like this. And I think the key point here is that it's less of sort of this sort of micro to macro specification of, I'm going to make a beak cell and an eye cell and a foot cell. And then once I have those, they'll come together and they'll make the next level, and sort of so on and so forth. I think it's more actually like a cappuccino, right? Where you certainly have two different materials, right? You have the froth milk and you have the espresso, and they have different material properties, but it's really about how they are really weaved together in space and time that gives the final duck pattern. There's not a moment where that's specified. So with that, I want to thank everybody. First and foremost, Amy Shire. We've been collaborating for about a decade now, and we co-direct the lab. So all of this work is a full collaboration with her. The rest of our lab at Rockefeller, both the past members and current members. And with that, I will be happy to take questions.
Questions?
I'm just curious, in like a hair placode, where you'll see invagination, like into the skin as opposed to a feather placode? Do you see like is that because of different material properties of those progenitors?
I mean, that's a great question. I mean, people like us would think about that, right? Yeah, like what would there could be. We haven't looked into that, but I guess the way we think about even answering that question is to think about sort of these sort of more supercellular mechanical properties and what could be happening to make it go in versus out.
Right. So in theory, Pearson could like tune his model such that he would get an invagination.
Yeah. And so there are like Evo-Devo type projects you could imagine, doing. Yeah. But you wouldn't be looking at sort of the change in the diffusion of a morphogen or something like that.
I can see how your holistic approach is intriguing, but if you're looking from an evolutionary point of view, I think the current concept is that animals arose from primitive choanoflagellates, where each individual cell had the properties of all the cells that we have distributed among many cell types in our current organisms, animals to date. So if that idea were to be right, then would we not still have to look very carefully as to which properties individual cells, as they develop, are being bestowed onto this particular cell type? Not every cell will be contractile, not every cell will be able to make cilia, for example.
Yeah. I mean, I think we have to keep thinking about kind of the molecular characterization. I think that's still important. I guess I'm just trying to say that we might actually make more sense of what's happening at the molecular level if we understood what's happening at the supercellular level that might be sort of giving those changes some sort of meaning or purpose. Like I think I think the supercellular level is perhaps this missing link between the organ level morphology that we all care about and what's happening at the cellular level. And it's too big of a leap to try to, and so it's not to say that the molecules aren't doing something. It's almost more like I think it might serve as like a phenomenological filter for what's happening at the molecular level. And I think that's sort of the next phase of the lab is now we want to go back and dig back into the molecular level and correlate it causally with what's happening at these higher levels.
So I'm curious, do you think that from material science and physics then, there are a collective set of emergent or behaviors that we haven't fit into biology and that's what we're missing? So how do we marry those two things together?
So is another way of saying that, like, is there more to biology than what we know in material science?
No, I
Or-
mean, like the idea of jamming, when you suddenly have cells together, you realize, hey, there's a polymer physics has really said, hey, this is a property, and then they realize that's what the embryo are doing. So are there a set of these collective behaviors that have been seen in physics and other studies that we can integrate into biology? Or do we really need to discover them from biology and then find how they apply to physical science?
I think it's the latter. I think we have a lot more. I think it's why a lot of physicists are moving into biology to characterize it, because they realize that quote unquote active matter has principles that are not present in polymer physics. So I actually think, because and I've talked to a lot of material scientists about this, that what they don't have in their systems as much is that the constituents don't change as the collective field changes, right? So when water forms a snowflake and that's self-organizing in a bottom-up way, the emerging architecture of the snowflake does not change the constituent nature of the water molecules, whereas that is what's happening in biological systems. And so that's actually hard to even model effectively. So the top-down aspect of it and the constituents evolving as the macro features emerge, that's sort of an exciting new area. And I think it's why biologists, especially the ones who can do good experiments, might be more valuable than the physicists for this.
I had another question, but so it's true that individual cells, no, they're doing their own business, but you mentioned the ECM, the extracellular matrix. Couldn't it be that this is the glue that binds everything together and that makes you're talking about the auto organizations of the cells, that it's I think a two-way discussion between the cells and the ECM, but the ECM in fact is the thing that binds everyone together. So wouldn't it be like a part of your answer?
Yeah, I think the matrix it's really critical, but these emergent properties are a combination of the cellular properties. It's a composite, right? So, but no, I think the matrix is essential, and I think in a lot of mechanobiology, a lot of the work is done on epithelial cells, where the matrix isn't as big of an issue. But I think, and this is something we're looking into. We're now looking into disease biology and looking into stroma, and how stroma might be something that's sort of also underappreciated and might, at the collective level, have a lot to do with disease.
Anyone else? Yes, there you go.
I have a question like regarding the length scale. So like when you mean by supracellular, like
Can you
the
speak up? We hardly
yes,
hear
so,
you.
okay, louder. So like, do you think like whether there are specific numbers of cells, like so what's the characteristic length scale related to like supracellular like properties? Like, do you think it's like cell type specific or?
I think about that a lot. I think the person who was really thinking about that a lot is Ignace Desai. I think he was thinking about that very carefully. If I were to guess, I think it's in the high hundreds to low thousands. And it's an interesting, I think it's a special number, and this is an issue in physics, which is physicists either, they want to coarse grain, so they just want to think about getting rid of all of the details of the individual molecules, but there's certain special numbers where you can't fully coarse grain. So I think that's what's tricky about this supracellular level, which is that there are these emerging properties, but you can't fully coarse grain and forget about the cellular details. But that's important because that's what links it to the genome, I think. And so I think that's the exciting frontier, right? Which is like, what are the sub features of the individual cells you don't want to fully coarse grain? And how are they linking to molecular features? I think that's kind of the exciting question.
Right. So I guess with this, we can.
Thank you. Thank you.